Hello everyone. In my previous two videos, I discussed about type 1 hypersensitivity reaction which is mediated by antibody IgE. Now in this video, I am going to discuss about type 2 hypersensitivity which is also mediated by antibodies like IgG or IgM. The major difference between type 1 hypersensitivity and type 2 hypersensitivity reaction is that in case of type 1, IgE antibody play important role, while in case of type 2, IgG or IgM antibody is involved. Then in case of type 1, IgE antibody is stationary or it is present attached to the mast cell or blood basophil, while in case of type 2 hypersensitivity, the antibodies are freely circulating inside the blood. Now, generally, type 2 hypersensitivity reaction is occur due to two different types of antigen, either intrinsic antigen or extrinsic antigen. In this video, I am going to discuss detailed mechanisms by which intrinsic antigen can induce the type 2 hypersensitivity, as well as how exactly extrinsic antigen induce type 2 hypersensitivity reaction. What is intrinsic antigen? These are the self antigen which is present naturally in the host or inside the body, and generally there are three important type of intrinsic antigen, like antigens which is present on the cell surface, then antigen which is present in the matrix, and receptor antigen. These are the antigen which is nothing but the receptor, like hormone receptors. Okay, while the extrinsic antigens are the non-self antigen which is generally enter inside the body from atmosphere, and there are three important reactions which lead to type 2 hypersensitivity with the help of extrinsic antigen. These are blood transfusion reaction, then hemolytic disease of newborn, HDN, and the reaction which is induced by drug metabolites. Then after discussing intrinsic and extrinsic antigen, I'm going to shift on how exactly antigen antibody reaction occur in type 2 hypersensitivity and how antibody destroy the target cell or the antigen. This happen with the help of opsonization and phagocytosis, or by complement activation, and by ADCC, that is antibody dependent cell mediated cytotoxicity. So first I'm going to start with intrinsic antigen. We know that hypersensitivity reactions are the inappropriate excessive reaction of the immune system that lead to damage to the host cell or host organ. Okay, in case of type 1 hypersensitivity, important role is played by mast cell and IgE antibody. After IgE antibody get attached to the mast cell, degranulation occur which release active mediators of the type 1 hypersensitivity like histamine, prostaglandin, which ultimately lead to contraction of the respiratory tract that lead to asthma, then itching and many other symptoms of hypersensitivity. Now in this case, in case of type 2 hypersensitivity, first is intrinsic antigen or self antigen. Three different type of self antigen can induce type 2 antibody mediated hypersensitivity. Immune response to extrinsic or foreign antigen is feasible, okay, but why immune system act on the self antigen? Because the reaction occur which is autoimmunity. The reaction of the immune system against self antigen is nothing but the autoimmunity. I am going to make separate video lecture on this topic, that is autoimmunity. But some self antigen like antigen on the cell can also induce autoimmune reaction that lead to type 2 hypersensitivity. Okay, so first is antigen on the cell. Cell like red blood cell and platelets which is present in the blood. Okay, and this RBC have number of different type of antigen on its surface. Similar case with the platelets. Okay, and sometime autoantibodies are produced against the RBC antigens or platelet antigen, and the antibody get attached to the RBC and platelet, which lead to rapid degradation or rapid lysis of the RBC and platelet. Due to decrease the number of RBC, condition occur called autoimmune anemia. Okay, and due to degradation or destruction of the platelet by autoantibody, thrombocytopenia is observed. So both this condition, autoimmune anemia while thrombocytopenia, is nothing but the reaction of type 2 hypersensitivity. Okay, and that happened due to antigen present on the RBC and platelets. Okay, and these antigen are self antigen. Then the next example of the self antigen is antigen on the matrix. We know that human body having 4 different type of tissue like muscular or nervous tissue, muscular tissue, connective tissue and epithelial tissue, and tissue are made up of cells. Inside the tissue, the cells are joined together with the help of matrix. It is nothing but the glue like substance which play important role of holding different type of cell together in the tissue, as well as this matrix is involved in providing nourishment or nutrient to the tissue cell, and this matrix also play important role in waste material decomposition or deposition. Okay, now this matrix have antigen. Okay, and this antigen of the matrix sometime recognized by the antibody present in the body, which lead to disease like good pasture syndrome. Okay, this is also example of type 2 hypersensitivity. In good pasture syndrome, some antigens which is present in the glomerulus matrix of the kidney or some antigen which is present in the alveoli sac of the lungs got attacked by self antibody or autoantibody, and due to attack of this organ like kidney, glomerulus and alveoli of the lungs, necrosis and hemorrhage occur in the kidney and lungs. Okay, this is called as good pasture syndrome, which is nothing but the type 2 hypersensitivity reaction. The third type of a self antigen which lead to type 2 hypersensitivity is receptor antigen. We know that various type of hormonal receptor antigen is present in the human body. That means these are the receptor which is present on the cell that recognize presence of hormone. Okay, this receptor mainly get attached to the hormone and then the signal transduction process occur. But sometimes some antibody get attached to the receptor which is generally made for hormone. Okay, and that lead to type 2 hypersensitivity. The example of the receptor antigen type 2 hypersensitivity is Myasthenia gravis. We know that neurons generally have gap between two neurons called synapse, and signal transduction occur from presynaptic neuron to postsynaptic neuron by neurotransmitter chemicals. Okay, for example, from presynaptic neuron, acetylcholine chemical is released, that is neurotransmitter is released, and this acetylcholine get attached to postsynaptic neuron on the spatial receptor. And after attachment of acetylcholine from presynaptic neuron to postsynaptic neuron, successful transmission of the signal occur. Okay, that means signal can be transferred from this neuron to this neuron with the help of acetylcholine. But sometimes in case of Myasthenia gravis, some autoantibodies are produced against the acetylcholine receptor present on postsynaptic neuron, and this antibody get attached on the receptors. This antibody block the receptor of acetylcholine, and due to which no longer this acetylcholine can transfer the signal from the synapse from presynaptic neuron to postsynaptic neuron. Signal transmission processes get inhibited that lead to condition called Myasthenia gravis. So this is all about the intrinsic or self antigens that ultimately cause type 2 hypersensitivity. Okay, and the example of self antigen is antigen on the surface of RBC and platelets, then antigen on the matrix surface which lead to good pasture syndrome, and antigen which is nothing but the receptor of the hormones and neurotransmitter like acetylcholine. Example is Myasthenia gravis. Then next type of antigen that lead to type 2 hypersensitivity is extrinsic antigen or foreign antigen or non-self antigen. Okay, the first example of this type of reaction is blood transfusion. We know that according to ABO blood group system proposed by Karl Landster, there are 4 different blood groups found in the human, like A, B, AB and O. The person with A blood group have A antigen on the surface of RBC, while the plasma contain antibody B. Okay, remember this thing, the person with A blood group have A antigen on the surface of RBC, while antibody B. Then person with a B blood group have B antigen on the surface of RBC, while the blood contain antibody A. Then AB blood group person have both the antigen A and B on the RBC, while no antibody is found. Okay, while in case of O blood group, antigen is absent on the RBC, but both the type of antibody is present in the blood. Now, if by mistake the wrong transfusion reaction is occur, that means a person with a blood group A is transfused with B blood. Okay, then B blood group person blood. Okay, in that case, now the person with A blood group have anti-B antibodies, and this anti-B antibody will going to attack on the B antigen present on the B blood group RBC, and due to which excessive hemolysis occur inside the recipient person's blood or inside the recipient person's body, that lead to damage of the RBCs. Okay, and due to excessive damage of the RBCs, high amount of hemoglobin is released inside the blood. Okay, and sometimes this hemoglobin get converted into bilirubin, and high concentration of the bilirubin is highly toxic to that particular person. As well as due to high concentration of the hemoglobin in the blood, some hemoglobin can also found in the urine. Okay, this is the medical symptoms that occur due to wrong transfusion. High concentration of hemoglobin in the plasma, then sometime hemoglobin is also found in the urine, and bilirubin is produced inside the body. The symptoms include chest pain, nausea, chill, fever, okay, and excessive agglutination reaction in the blood vessel. Only treatment for this type of wrong transfusion reaction is avoiding or terminating the transfusion as early as possible. Okay, the reaction occur due to wrong transfusion is the example of type 2 hypersensitivity. Okay, this is all about first, that is blood transfusion reaction. The second extrinsic antigen example is HDN, that is hemolytic disease of newborn. Here you can see the condition occur when mother is Rh negative while the fetus is Rh positive. Okay, if the mother is Rh negative, any blood group, A negative, B negative, AB negative or O negative. Okay, and if at the time of first pregnancy, if the fetus is Rh positive, any blood group but having Rh antigen, in that case no problem occur during the pregnancy, but at the time of delivery the blood is passed from umbilical cord from fetus to mother. That means RBC, Rh positive RBC is transferred from fetus to mother at the time of first delivery. The mother body gets sensitized due to this Rh positive RBC. The mother body immediately produced antibody like IgG and some memory cell. Okay, so no severe condition occur during the first pregnancy, but the mother body gets sensitized and memory cells are produced. Now in the second pregnancy, if child is again Rh positive, in that case mother body already have IgG antibodies and memory cell. Okay, now this IgG antibody can easily pass through the placenta, okay, and enter into the blood of fetus, that ultimately lead to hemolysis. Okay, and this condition occur during the second pregnancy is called hemolytic disease of newborn. To avoid this condition during the second pregnancy, after first pregnancy, mother is injected with a drug called Rogam. That drug avoid sensitization of the mother body, formation of memory cell and IgG G antibody. Okay, Rogam is the effective drug that avoid problem that cause at the time of second pregnancy. Okay, so this is all about the HDN or hemolytic disease of newborn, which is also an example of type 2 hypersensitivity. The last extrinsic antigen which lead to this reaction is drug metabolites. Various drugs that can cause type 2 hypersensitivity. Well-known example is penicillin. When we take penicillin, it is metabolized in the liver, and the metabolite or the fragment which is produced due to metabolism of the penicillin get deposited on the surface of the self cell. And these metabolites of the penicillin can be act as a extrinsic antigen. Immune response is built by the immune system against this metabolite, and by clearing at the time of clearance of this metabolite, some self cell may get damaged. Okay, this is the drug metabolite reaction. Okay, and this also example of type 2 hypersensitivity. So three important self antigen and three important non-self antigen mainly cause type 2 hypersensitivity. Now, exactly which mechanism is employed by the antibody to attack self or non-self antigen? Okay, these mechanisms are divided into three main types, that is opsonization and phagocytosis, complement activation, and ADCC. Okay, what is opsonization? It is the condition in which target cell or infected cell, okay, is first coated with antibodies, and after binding of the antibodies to the epitopes or the antigen present on the target cell, now the Fc portion of the antibodies are easily recognized by phagocytic cells like macrophages or microphages. And after binding to the antibody Fc portion, now this phagocytic cell can easily engulf this target cell, which lead to phagocytosis and death of the cell. This type of reaction mainly occur at the time of type 2 hypersensitivity when immune cell use antibodies to kill target cell. Okay, this is called as opsonization and phagocytosis. Then sometime when any self or non-self antigen present in the body, antibody like IgG or IgM get attached to the antigen, which lead to initiation of the complement pathway. There are three important pathways of the complement system, like classical pathway, alternative pathway, and lectin pathway, out of which antigen antibody complex activate classical pathway that ultimately activate complement protein, and which finally end with the killing of the antigen. Okay, this is also one of the mechanism used by the immune system to kill antigen with the help of antibody. While the third and last mechanism is antibody dependent cell mediated cytotoxicity, that is ADCC. What is ADCC? When any target cell get attached with the antibodies, then natural killer cell or effector cell can easily recognize this target cell, and instead of phagocytosis, after recognition, natural killer cell produce several chemicals, several active mediator like cytokines, interleukins, okay, tumor necrosis factors, and now these cytokines directly attack on the target cell, which lead to death or damage to the target cell. This is called as antibody dependent cell mediated cytotoxicity. So in this lecture, I discuss in detail about type 2 hypersensitivity, which is mediated by antibody, mainly IgG and IgM. This type of hypersensitivity reaction is quite different from type 1, okay, because type 1 is mainly induced by IgM antibody. Okay, generally two different type of antigen is involved in type 2 hypersensitivity, that is self antigen and non-self or foreign antigen. Self antigen include antigen present on the RBC and platelet, which lead to autoimmune anemia and thrombocytopenia. Then antigens of the nephrons of the kidney and alveoli of the lungs, which is targeted in good pasture syndrome, while the antigens, which is nothing but the receptor, example is Myasthenia gravis. Then extrinsic or foreign antigens is blood group antigen, wrong blood transfusion reaction. Then the condition which occur if the mother is Rh negative and fetus is Rh positive, it is called as hemolytic disease of newborn. And the condition which occur due to drug like penicillin when metabolized, and this metabolite of the drug lead to type 2 hypersensitivity. These three are the examples of foreign or non-self antigen. Finally, we discussed different mechanisms by which antigen is killed by antibody. Okay, and the mechanism is opsonization and phagocytosis, killing by activation of the complement pathway, and last is ADCC, that is antibody dependent cell mediated cytotoxicity. In my next lecture, I'm going to start with type 3 hypersensitivity. Thank you.